we are here, right? Yeah, your workshop. Yeah, yeah, I care. In fact, I'm going to, hopefully with time, I'm going to talk about my work with Mandar. Yeah, yeah. I think I'll start.
Okay, so welcome back to the workshop. We're on day 8. So hope you've been enjoying it so far. Today we are delighted to once again have Professor Shashithara back with us, and he will be giving his second lecture on more questions related to development. So please go ahead.
Thanks. So happy to be back. I'm quite impressed that 2 weeks later people are enthusiastic. I saw many students here already by 9:15 sitting here and discussing. Hi. So in my first lecture on the very first day of this workshop, I was talking about the first principles of development, right? Starting from a single cell embryo, in fact, the process of development starts, prior to fertilization. In fact, time t is equal to 0 is not fertilization, it's even prior to fertilization. But whatever, we're talking about how from a single cell you get large multicellular, very complex organisms such as human with variety of different kinds of organization in space, right? And so you start with almost no information, no polarity, no access information. Then every cell in that space in the developing field is assigned a specific identity, certain values, and values are determined with the help of certain kinds of proteins which we call as morphogens, and how those morphogens are expressed, the gradient is established, and how they confer identity to individual cells. That's what we discussed. Also sort of ended with some open questions. That's what I guess you have discussed subsequently. What is the robustness of the gradient, and what is the time scale of gradient formation, how gradient, in terms of variations, and how different versions of the gradient are used during development, and what are different other developmental mechanisms. One is morphogenic gradient, another is, we discussed, I'll come back to that again, self-organization, through cell-cell interaction, recruitment, lateral inhibition, and variety of different types of developmental mechanisms are operated during development to generate such complex system. Today I thought a little bit about EODO, what's called evolutionary developmental biology, right? It's, while we say very different developmental mechanisms are highly conserved across even plants and animal kingdom, any multicellular organism, there are certain kinds of fundamental mechanisms are conserved. One is because they had to be together, these cells. Second one is cells of a particular type form a tissue, and they had to be together. And how do they get separated? What kind of cell adhesion properties or other mechanical properties of the cells are coming together to establish those tissues and organs and so forth. But if they are so well conserved from across, but we still see diversity in morphology, right? And that's one thing what we thought we'll discuss today. In addition, some other questions. So fundamental to the entire living system is the chemistry of life, which I'm sure all of you are aware of it. In this chemistry of life, the most important aspect of it is the how the DNA or genetic information is copied, right, every time. And if the copying mechanism is 100% error proof, there wouldn't have been life in the even a very first day after the origin of life. The reason we have survived for 3.75 billion years is because there is some what called mismatch pairing happens during copying mechanism, right. So, instead of ATGC, sometimes you do get AC or GT base pairs, although they are not chemically very stable, but they do get incorporated once in a while depending on certain kinds of concentration of nucleotides during the copying. And although it is a stochastic phenomena, there is no any mechanism by which this kind of mispair or DNA copying errors are removed during replication, but some of them are retained, right. The retained, sometimes if they are also even enhanced, magnified to a specific mechanism for generating more variations. Without these variations, there would not have been ancestral life, simply because the environment is continuously changing. To change to adapt to the changing environment, organisms need to have new genetic variations, and those genetic variations are generated through this copying mechanism during the DNA replication. Now, what happens when there is a change in the DNA, the genetic information, there will be some structural changes. It could be catalytic RNA or the protein. Those structural changes are the ones which may or may not have a new function, or some existing function may be improvised or compromised, and depending on that situation, a new function may emerge, right. So, this is how the evolution sort of progresses, and this new function could be the substrate for natural selection. If that new function is favorable for the so-called fitness of the organism in the population, it continues, spreads at higher frequency, otherwise it sort of spreads at lower frequency, or sometimes it may completely get eliminated if it is highly deleterious. And this is how it functions, but it is not straightforward, it is not linear relationship, it is pretty non-linear. And just to give you an example of non-linearity, a qualitative example of a non-linearity is this one, right. All of you know about the famous example of sickle cell hemoglobin. It's just one nucleotide change in the 7th codon, leading from change the glutamine amino acid to valine, right. Now, the so-called within quotes, when I say normal, it is always within quote, normal hemoglobin packs very nicely to disc-shaped RBCs, whereas this variant of hemoglobin is not so well packed, it forms sickle cell shaped RBCs, right. So, these RBCs morphologically very different just because of one amino acid difference in the protein, right. Heterozygous individuals, that means one copy of normal so-called normal and one sickle cell hemoglobin individuals at higher altitude have a problem of oxygen exchange. So, they have sickle cell anemia phenotype, right, which you can call as disease. At mean sea level, where oxygen levels are pretty high, concentration is pretty good, they do not have any major impact on their physiology, right. But assume that if the malarial parasite, which has co-evolved with many animals from birds to all mammals, this has co-evolved to invade so-called normal RBCs. Typically in co-evolution, the to host pathogen or symbiotically, whatever is the relationship, the evolution is always favorable in terms of numbers to whatever is the highest frequency in the population. So, typically malarial parasite has evolved to invade the disc-shaped RBCs. They cannot enter so easily the sickle cell shaped RBCs, right. So, in the process what happens is, if the mean sea level where none of them have any major impact because oxygen level is high, but if there is also malaria endemic, then these individuals are susceptible to malaria, where these are resistant to malaria, right. So, what is fitness in terms of an allelic function is also context specific, it is not in absolute terms. Because of this, there are a variety of different kinds of functions may emerge, phenotypes may emerge, variations may emerge, both morphological or physiological, and a combination of both, leading to this kind of biodiversity that we see, right? But still, all our explanation to evolution is what is called explanation to microevolution patterns. Microevolution patterns, we only talk about new variations in the genetic material and how it spreads in the population. But it will never sort of explain so easily the major morphological differences even among the closely related species, right? And that's where the question of evo-devo comes, right? And then something which we are going to discuss, and we discuss, I'm sure many others speakers who have come here also have discussed morphogen gradients, signaling pathways, some of the master regulatory genes, and so forth, as part of developmental mechanism. Then evolution, obviously, if it operates on any one of these things, and there would be changes in the developmental mechanisms, and that's one of the things we want to discuss. I also gave you one example in my first lecture, although I didn't elaborate much. For example, when morphogen gradient in Drosophila, a protein is sort of diffuses through the cytoplasm because it's a single cell, although it's multinucleate, and because it's a protein is nuclear protein, when it enters into the nucleus, and from one end to the other end of the embryo along the longitudinal axis, different nucleus, sort of takes up different amounts of that same protein, and that's how a morphogen gradient is established. Whereas in case of vertebrates, the gradient is established after cells have already formed, right? And then that means any factor which establishes as a gradient has to go through a cell membrane. It's not simply the gradient which is basically passing through only the cytoplasm, right? So that's some variations the mechanism do operate, but conceptually it is still a gradient. I also mentioned typically what is a eukaryotic gene structure and how very different features of a gene are involved in ensuring that the coded information is expressed in specific location, in specific time, specific location, and at specific quantities, right? Now, if you look at simply based on the gene expression, if you sort of, and a reductionist approach to development is spatiotemporal regulation of gene expression, leading to from a single cell to multiple cell complex system, where different cell takes up different identities, although they have the same genome, right? And that's the sort of logic in which we started, right? Now, in the context of evolution, there can be multiple things. For example, there is a one particular family of genes, let's say, or one particular gene, but with a specific function, and through duplication and divergence, it may evolve to multiple different types of functions. And each time when there is a duplication divergence, some new feature may be added to the organism. But how it is acquired, how it gets selected, is still an open question that we need to discuss. Or there can be change in the gene expression pattern. Let's say I specifying gene in human, it's expressed here and nowhere else is expressed, right? So a gene gets activated in the place where the eyes should be formed, at the same time it is repressed in other places, right? But now you change the gene expression pattern. So instead of eyes here, eyes can be somewhere here. You can simply change the location where the eyes are formed by changing the gene expression pattern. Or you can have, let's say, instead of 2 places where eyes are formed and genes are expressed, you make it in 4 places, there can be 4 eyes, right? So there are specific examples. I mean, eye is a very wrong example I'm giving. For example, arthropods, there are 2 types, right? Then the insects have 6 legs. Non-insect arthropods, some of like spiders have 8 legs. Millipedes and centipedes even multiple legs. Now, the leg features, what genes specify legs, could be the same gene, is simply because gene expression is changed, giving rise to multiple legs. I'll give you those examples. Or same gene in different lineages have slightly new biochemical properties, leading to some new features in the morphology, right? Or if it is a transcription factor, or there can be 2 proteins come together and collaboratively act which may be different in different lineages, leading to morphological divergence. Or if it is a transcription factor, it binds to certain downstream genes, and the gene in the cis regulatory element, and that gene expression is leading to a morphological feature. But those downstream genes, they change, either protein expression changes or the way the DNA binds the transcription factor, those elements may change. That also leads to change in the morphology. So just one example of duplication divergence. In insects like Drosophila, there are 8 Hox proteins. I told you what is the Hox protein last time. There is basically a family of master regulatory genes, and those are the ones which require to make region specific or domain specific features along the body axis, particularly along the anterior posterior axis, right? Now there are 8 of them, but through a duplication divergence, we know now through a variety of different bioinformatics and experimental evidence, there are these 44 genes that are present in mammals. They are the products of the original 8 Hox genes that are present in insects. And along the body axis, there are more features in more complex organisms which have more Hox genes. So there's clearly suggests that how complexity may have emerged, simply evolved by duplication and divergence of Hox proteins. In fact, there is also a linearity in how they have evolved from one end of the organism to the other end. In fact, the most of the new features are added to the posterior end of the body along the axis. So if, for example, some of these genes which are expressed here, then you get more and more genes gets added here, and new features are emerging. Right? Gene expression pattern. Here is a fantastic example. There are multiple examples, including millipedes and centipedes. I'll just use this. It's a very dramatic example, although there is really no direct experimental evidence for this, except some observational evidence. Unlike in Drosophila, where a single Hox protein is required to make a specific feature, in vertebrates, because of the complexity perhaps, some of the Hox proteins also collaborate to specify more complex structures. Hox C4 and C5 are required to specify the forelimb, right? Like in mammals, the 2 arms, or sorry, in humans, 2 arms. Mammals, let's say front legs. This is in case of any vertebrate with 4-legged one. C6 and C8 are, C9, 10 and 11, there are 3 Hox proteins collaborate to specify the hind limb, right? And C6 and C8 is required to specify the distance between the 2 limbs, the fore and the hind limbs, right? Depending on the species, you all know that there is a difference in the distance between the forelimb and the hind limb, right? And for example, this is in case of birds, where although forelimbs are modified into wings at some point during evolution, basically you call it a forelimb and this hind limb. Hind limbs are the legs in the birds, and C6 and C8 are expressed like this. So observation is such that the posteriorly expressing Hox proteins are dominant over the anterior ones. So because C6 and C8 is expressed here, right, even if C4 and C5 get expressed here, it cannot function. But C6 and C8 gets expressed in this domain also, even if these are expressed, but C6 and C8 functions dominates over C4 and C5 function. Similarly, 9, 10 and 11 function, if you put it here, it will dominate over 6 and C8 or C4 and C5, but these 2, if they overexpress here, they will not be able to suppress the function of these 3 Hox proteins, right? And given these observations, people have also found out during early embryonic development, in case of, for example, birds, this is how it is. C6 and C8 are expressed to this level, so that forelimb and hind limb are kept with certain distance from each other. But in case of pythons, people have observed that C6 and C8 expression is further towards the anterior vertebrae and suppressing the formation of the forelimb. So two things happen. One is there are no forelimbs. At the same time, the so-called flank, the region between the two limbs, is further extended. So what will happen is you have a long body, but only two legs at the back of the of the body, right, at the most posterior one. So it's like a long haul truck with only two wheels at the back and no wheels in the front. So only way it can sort of survive, it's just to collapse on its belly on the floor. Now, one assumption is when it got extended like this, the body is stretched with only two legs, and these two legs will not have any function now, because with two legs it cannot move around. So it will only move around by crawling, right? And then if there is no function to these two limbs, they can accumulate any kind of variations because there is no impact on the function of the hind limb. Then it becomes rudimentary. Indeed, pythons have two rudimentary legs at the posterior part of the body, whereas the much of it is without limbs in the front. Now, this is a very simplistic explanation how the morphology could have evolved, but it will still not explain the selection, because there could be a time where, how does it survive, right? Until the time when it has to evolve certain other features of muscle movement and everything for the movement and from walking towards the crawling and kind of stuff. Selection is, and then the selection, right? And there are a variety of complexities associated with this, but this could be the beginning of any evolution of any new morphological features, right? There's other one. For example, in case of insects, which have evolved much later compared to other arthropods, insects have only 6 legs, one pair each along on the 3 thoracic segment. The body is divided into 3 parts: the head, the thorax, and the abdomen. And whereas, for example, if you look at other non-insect arthropods, there are legs not only in the thorax, also in the abdominal segments, right? Now, if you look at the protein, which is a particular protein called UBX, right? Don't worry about it. It has what we call DNA binding domain, right? Which is, and then you have a certain kinds of N-terminal and C-terminal regions, right? And if you take the UBX from very different organisms, these 3 are insects. First one is Drosophila, second is Tribolium, third is a butterfly, and the fourth one is a shrimp UBX. And if you look at these 4, and there is, while homeodomain region is highly conserved, irrespective of whether it's an insect or a non-insect arthropod, if you look at the C-terminal domain, there are some amino acids which are very different compared to all insect UBX. All insect UBX have polyalanine tail at the C-terminus, whereas the non-insect one doesn't have that kind of a polyalanine, but it has some kind of a serine threonine, which can be phosphorylated. It doesn't matter for this discussion. But anyway, they are different. So people have shown that UBX is required to suppress abdominal leg, right? So insects have only 3 pairs, but all 3 pairs are in the thoracic, but there are no abdominal legs, right? So if UBX is overexpressed in the thoracic regions where legs are formed, you can see suppression of one. There is, don't worry about which gene it is. There is a gene which is specific to the beginning of specification of the leg identity in these cells, and which is shown with the help of a GFP marker. And if you overexpress UBX in these cells, you see that expression pattern is, expression levels is coming down, suggesting that limb or leg formation is being suppressed. If you take Tribolium UBX, put it in Drosophila, right, overexpressed like this, you see that the leg is being suppressed, right? But if you take another insect arthropod UBX overexpressed in T1, T2, or T3, you see that there is no suppression. That means it has lost the ability to suppress, or doesn't have the ability to suppress. Not that it has lost, it doesn't have the ability to suppress limb formation. Now, if you make chimeric protein, take the N-terminus of the non-insect arthropod UBX, just take only the C-terminus of insect UBX and make a chimeric, Now this protein can suppress limb development. You can see the levels of GFP coming down, suggesting that the insect UBX has acquired new function because of these protein sequence changes that has evolved with time, and that acquisition of new feature has made the body plan completely different, right? One with multiple legs along the body axis, another is only 3 pairs of legs in the thoracic segment, right? And so you can see how one can explain evolution of morphological features, which basically, while the basic developmental mechanisms are conserved, but you can still see some new features can evolve. So not necessarily that entirely new development mechanism itself has to evolve. Certain minor changes in the existing developmental mechanism, some gene expression or gene function can change to new features. Now, if you look at the other factors, what other mechanisms can operate. Here is one example which we have worked on it. Earlier, people like Ed Lewis and others have shown the function of UBX. I already spoke about this earlier. UBX is required to make the halteres in the third thoracic segment, and UBX does so by suppressing wing development, and UBX is sufficient to make halter, right? This is the normal fly, this is the UBX loss of function, this is UBX gain of function, right? Now, if you look at the certain groups of insect, what called holometabolous insect, these are the insects which undergo what's called metamorphosis. There are 2 stages in their life cycle. Early during embryonic development, they give rise to a larva, and later, after certain stages of larval development, these larvae pupate, and inside the pupa, the larval tissues are lysed except the nervous system, and new tissues are formed, which basically makes the adult structures. And this change, we call it as metamorphosis. And if you look at amongst the insect which have these kind of metamorphosis process, these are the what called dragonflies, which commonly known as the dragonflies. They have forewing and hindwing which are pretty identical in type, size, shape. Even there are decorative patch can be identical between the fore and the hind wing. Then there are 4 classes which have some divergence. In case of bees, honeybees and others, forewing and hindwing are very similar, except that hindwing is somewhat smaller. In case of beetles, the forewing is modified into thick structure called elytra, whereas the hindwing is retained as flight appendage. If you look at the lepidopterans, butterflies, moths and others, you have forewing and hindwing are different in terms of size, shape and decorative patches, but still functionally they are wings, right? They are flight appendages. And the dipterans, like coleopterans, have another extreme changes where the hind wing is modified into 2 balancing organs called halteres. They are not direct flight appendages, so only 1 pair of wings act as a flight appendages. Similarly, in coleopterans, only the hind wing functions as a flight appendage. Now, yeah. Yeah. That's true.
Any of these?
Yeah. I think I may have, no, I don't have for dragonflies here.
Okay.
Dragonflies are the earliest ones, and then they come here in the phylogenetic lineage. And then there are, this is the divergence.
And they form a separate group altogether?
Separate group.
Okay.
So if you look at, some of the ways, gene expression, gene level evolution would change to morphological evolution, right? One is of course change in the number. We know that all insects have only 8 Hox proteins, and as far as the teeth, third thoracic appendage formation, it's a UBX which is involved, so there is no difference. Then change is the domain of gene expression. We looked at it, and we found out that is very interestingly, in case of hymenopteran, UB is expressed both in the forewing and the hind wing. In case of coleopteran, it is expressed partially in the forewing and very highly expressed, intensely expressed in the hind wing. And in case of lepidopterans, it is expressed partially in the forewing, and when you say partially expressed, there is a particular layer of cells called peripodial membrane which is expressed, which is not directly involved in wing development. It is expressed in case of these ones, whereas in all of these cases, coleopterans, lepidopterans and dipterans, UBA is expressed in the hind wing, also in case of hymenopteran. That means UBX expression per se is still present, because one of the prediction is if UBX expressed, it will make halter. That means UBX may not be expressed here or expressed here or expressed here or not expressed here, which is not true, right? So gene expression is not necessarily the one. Is it the protein which is different in these ones? By the way, there are 350 million years of evolution just for these four, from Odonata to the further divergence of these four groups, over 350 million years. It's not short time. So all kinds of changes could have happened, right? So what we did was to see whether UBX at the protein level has evolved. One way to do this is by complementary experiment. If you remove UBX, you get a wing-like structure, right, in the forewing or in the third thoracic segment. Now, if you put an exogenous UBX, and you can suppress the loss of function of UBX, right? This is called complementary complementation experiment. It can be done in yeast and on all kinds of system, in bacteria, everywhere. Now, if you put Drosophila UBX back through an exogenous system, you can see the normal halter is back, right? So that means this experiment basically is to show that it works, right? Because it's the same UBX. What is lost is also Drosophila UBX. What you have put back is also Drosophila UBX, although through a transgenic system. Now, if you take Tribolium UBX or Bombyx UBX, you can still get the halter back. Although UBX is present in the T3 of Tribolium or in Bombyx. Bombyx is the silk moth, which is the lepidopteran example, right? They don't make halter, they make wing. But when you take the UBX from those insects and put it in T3 in Drosophila, what you get is a halter. That means events downstream of UBX must have evolved and not at the UBX level. That's what it means. So to do that, what we did was to do a chip experiment, chromatin immunohistochemistry experiment. For example, it is amongst the non-biologists here. If you can look at all the regions of the genome where a specific transcription factor is binding by immunoprecipitating the transcription factor along with the DNA on which it is bound, and then you sequence the DNA, right? And then map to the genome and see where these sequences are. And also you can identify what kind of regulatory elements the UBX binds. So we found out that there are small variations in the way UBX binds in Drosophila versus Apis. It's just one example. And we can do Drosophila versus other insects also. This is Apis is the honeybee. Now, in case of UBX, there is you just look at the core of where the UBX binds in the DNA. It is thymidine, then 3-alanine, ATP, and then one more T, right? T-AAT. In case of Apis, it is TTAAT, or you can call it a TAAT, right? Just because these 4-mer, if you look at it, and this is a 5-mer and this is a 4-mer, right? There's a small difference, one nucleotide difference you can call, right? Now, we experimentally tested across some 2-3 different genes. One of them I'm just showing as an example. Here is a gene called vestigial, which is required to make wing development. And what UBX does is to suppress this gene expression in the halter so that wing development is suppressed, right? The way I showed about the leg development, a specific gene is expressed to induce leg development. By you expressing UBX, you can suppress that one and suppress leg development. Here too, the suppression of this particular gene means the wing development is suppressed. Although you may not see this at this level, it's a very early stages during development. Later in the adult you can make out, right? Loss of vestigial is wingless. Now, if you take just the regulatory region of this gene, put it a reporter gene called like a GFP, GFP is expressed in the developing wing, but is not expressed in the developing halter, because halter is not a wing, and UBX is required to suppress this expression. But you take the regulatory element of the same gene, put it along with the GFP reporter gene, and take it from apis genome, the honeybee genome. What you see is it is expressed both in the wing as well as in the halter. There are two interesting things, right? One is, of course, because there is a wing in the T3 in the honeybee, it has to make the wing, so this gene cannot be suppressed. So in a way it's expected. But more importantly, look at how well is conserved in the context of conservation of developmental mechanism. It is a 350 million year apart diverged two insects, one Drosophila, other Apis. That enhancer is just 100 or 200 base pair long enhancer can function like Drosophila enhancer, which is expressed in the wing, but because it is not responding to the presence of UBX, and it is still expressed in the halter, right? Now the question is how to make it responding to the UBX. As I told you, by the way, just the it's the same as T8T or A8TA is the same. It's just the other side, the complementary nucleotides. So what we did was to just change one nucleotide, right? Instead of making 2 T's, I here make it 3 T's. And UBX, earlier we have shown through biochemistry that UBX binds strongly if there are 3 T's or 3 A's between 2 A's, right? And by changing in the transgenic animals here, you can now suppress UBX vestigial expression in the developing halter, suggesting that what has evolved is these regulatory sequences on the gene which are making wing are required to make the wing, where UBX binds and suppresses that. Now UBX binds so strongly and suppress in Drosophila, but it cannot suppress in case of Apis, right? Although UBX can bind mildly, but it cannot suppress. Otherwise, you would not have pulled it down with the help of cheap experiment, right? This could be another evolutionary mechanism for morphological divergence. So another very dramatic example, this is work of Walter Gehring's lab in Basel. So, divergence of the downstream events. So I told you about eye development. There is a gene required for eye development. It is supposed to be expressed in where eyes are formed, and it should not be expressed where eyes are not supposed to be formed. But if you overexpress this gene, this is in case of Drosophila, you can see new eyes coming on the leg or virtually anywhere in the body, right? So you can have multiple eyed insect, but although not fully developed, because there are other parts of the body is not conducive for perception through these ones. But they do have all the features of an eye. They have photoreceptor neurons. If the light falls on this, those neurons fire, but no information goes to the brain because they are not connected to the brain. If you remember in the very first slide I was talking about, development is all about 3 aspects. One is positional information, diversification, formation of tissues and organs, particular size and shape, and physiological connection amongst all of these things. In the absence of physiological connection between these eyes and the brain, there is no perception through these eyes, but they are perfectly all right at the single cell level at eye-like structure, right, at every cell here, pigment cell or the photoreceptor cell. Sorry? Yeah, that's what I'm saying. You put the light there, it show the fire, those neurons fire.
Okay, and how do you actually induce those specific gene expression? I mean, what's the technique?
So the gene there are 2 ways of looking at it. One is this particular protein is called PAC6 protein. In Drosophila it's called ILS, and you can overexpress using specific regulatory sequence, or you can randomly overexpress anywhere in the body using certain flip FRT method. You can use a US PAC6, and the regulatory could be tubulin. It's just expressed in every cell, but usually it will die. But you can use it what's called a Galford system, which is under FRT, So this GAL4 randomly is expressed in different parts of the body. Wherever GAL4 expressed, UAS, PAX6 also gets expressed. Now, what Walter Gehering's lab did was, yeah.
So I read somewhere about the canalization of development. So basically, we cannot, like in the case of humans, there are basic wind pathway and all those things that only through that we can develop something, but we cannot develop anywhere. So how it is related
Yeah.
to that?
Good question. In fact, Evo Devo, the field itself is to see how this canalization is basically makes the whole system very robust, right? For example, in case of UBX mediated halter development, you can say there are multiple pathways are involved. So as though there's a single gene, right? You remove UBX, you get a wing, but there is no other example of what other genes you can remove to get a wing in the place of halter. It's because downstream of UBX, there are multiple pathways are upregulated or downregulated, but they are all sort of, canalized towards making a halter and by suppressing the wing development. So this makes the system very good, because by mistake, if any other genes are mutated, there is no reversal of the phenotype, right? And also the halter function remains as halter, both in terms of structure as well as its function. In case of IDE, interestingly, although there is a gene PAX6 which is required to position where the eye should be, but downstream, if there are at least 8 to 10 genes, they are also equally important to make the eye. But if you remove any of them, you can actually see loss of eye development, but you cannot make an ectopic eye, right? So for example, this particular protein, there are downstream, there are multiple genes that are required. You can see loss of eye development, but you cannot make ectopic eye using overexpression of those genes. The same thing with UBX. You can overexpress UBX in the wing, you can make wing into an halter. Removing UBX, you make halter to wing. But downstream, there are very different genes. Some, if you remove, you get both loss of wing and halter. Sometimes you see some distortion in the structure, but you cannot make an ectopic new structure. The idea of Evo Devo is to see how this well canalized, robust system, you can break it down, and then new features can evolve. That's why most of the example we are given is very preliminary beginning of Evo Devo field, where based on the gene expression you can predict what could have happened, but it's still not going to explain what is the complex evolutionary path it has taken. So the genes that are required for eye development in mouse or human in the early stage, where to position the eye irrespective of what kind of form of eye, the protein has similar kind of biochemical properties. It's a protein equivalent to PAX6, right, which is particular type of transcription factor. We will not get into the details of biochemistry of those proteins. So what Walter Gehering's lab have shown that you take PAX6 from mouse or human and put it in Drosophila eye, antennae. In the place of antennae, you are now seeing a small eye coming up, right? And this is the normal original eye, and this is the new ectopic eye. But this ectopic eye can be formed by overexpressing PAX6 from Drosophila or from human or from mouse, right? Now, what it suggests that early in the during evolutionary one, while more or less everything is retained, the biochemical properties is the same, where it is expressed is the same, but still what you see is this kind of eye versus this kind of eye, right, or this kind of eye. So some minor morphological difference between two mammals, or major morphological difference between a compound eye of an insect versus a retinal based this kind of eye, right? And these differences have evolved because of events downstream of this protein, which is the fundamental to make the eye in particular position along the body axis. Any discussion you want to have on this aspect where we discuss the evolution of developmental mechanisms or evolution of new morphological features? Because I thought I'll talk about this at the end of this workshop, because you must have already gone through very different developmental mechanisms, in different context. Yeah.
So Sri, I had two specific questions about what you said. So, in the Hox genes, how much variety is there within vertebrates and within insects? So you said that there are 44 in mammals and mouse, but
Yeah. So these 44, I showed you the right. It can be each one of them can be taken back to one of the 8 genes of the
Okay.
original insect UBX. Insect Hox proteins. These 44 are grouped into generally 4 groups, Hox 1, 2, 3, 4 groups. Within each group, there are multiple ones. In insect, there are basically 2 groups, Antenapedia group and UBX group. There is the anterior group and the posterior group, you can call it, right? Whereas here there are 4 groups, and each group has multiple members.
so all mammals have these 4
All
groups,
mammals have this 44.
I see. Okay, great. And for the UBX,
Even in I think in fish also you have 40 plus 44, I guess.
44.
Same 44.
I see. So there's nothing between these 2 groups and 4 groups.
No, there are.
Okay.
There are cases where there are, fewer. I think the highest number is mammals, that is 44, and then 8 in insect. There are smaller than 8. For example, yeast has only 1 Hox-like protein,
Okay.
whereas other C. elegans has, I think, fewer than 8. I think 3 or 4. Whereas I think if you look at some of the reptiles, they have fewer than this.
And the UBX, I don't know if I fully understood what you're saying, but is it possible in the fly that UBX is expressed, but some downstream mutation still causes wing development, or that's not possible?
So far, every kind of experiment people have done, we also have done. Saturated mutagenesis is the first one people tried, and not a single mutation has been able to transform
I see.
wing towards the hull, hull tail towards the wing. What we tried was called overexpression, genome, basically genome-wide overexpression experiment, where every Drosophila gene is overexpressed in the hull tail and see whether it gives a phenotype. We did it 2 kind of combination. One is in the normal genetic background, and there is UBX heterozygous, which is sensitive genetic background, where we saw some partial transformation towards hull tail to wing, but only the marginal bristles, pigmentation, or the size of the hull tail. But we couldn't see any flattening of the hull tail, or we couldn't see vein formation, such kind of phenotype, which is more wing-like, right?
Interesting.
Whereas in what's the other experiment? The chip experiment, which basically, again, we give all kinds of transcriptomics kind of a thing. Multiple genes are upregulated, downregulated. When you overexpress what is downregulated, you still only see some changes. We could never see a flattening or a vein formation. Flattening we could see only if 3 genes at the same time are, overexpressed. Right.
Interesting. Okay, thanks. Actually, I also had a broader question, which is that could you comment on, during the whole developmental process, are there times which are sort of more susceptible to change versus other times? I mean, is any of this?
So I'll come to that in a way in one context in our work, but it's true. So the blueprint formation, we actually in the very first lecture were talking about the molecular changes to start with, then precede the morphological changes, right? So you have morphogen gradient, and very different genes are expressed depending on how much of the morphogen protein is present in a given cell, and very different downstream genes are expressed. We still do not see any morphological changes. During that time, you can change the gene expression, you see morphological changes. But after some time, when morphological changes are already initiated, in a way the position the positional identity of the cells is fixed. By overexpressing the gene, you cannot do something. Same thing, for example, easier best way to example is the ectopic eye development. Or ectopic leg development, antennapedia, or this experiment I showed you, ectopic eye development. You can do only up to certain stage in the larval development overexpress. Even at very late in the larval stage or early during pupal stage, if overexpress, nothing happens, because there are already irreversible identity is already acquired by those cells. Then you cannot change. Is that there is a temporal sequence is very, very important.
So I have a question about these Hox genes. So you mentioned that they are present in a sequence, and this sequence corresponds to the body segments they act on, right? So they are linearly present, and each of these genes correspond to the body segment. So why is that necessary? Such a sequence necessary? Like.
So you have to hold this microphone.
Oh yeah.
Repeat
sorry.
again.
So these Hox genes, you mentioned that they are present in a sequence.
Yeah.
And each of and this sequence corresponds to different body parts in the developing embryos. So why is it necessary that the genes have to be present in a sequence in the genome?
That's how it has evolved, because of duplication and divergence.
Okay.
Very for example, in the initial, if you look at in the early evolution of morphological features in the animal body, other than bilateral symmetry and bilateral asymmetry evolved much later in vertebrates, right? Most animals have bilaterally symmetrical. So there is, if you can see here, the evolution of that happened much later. So any gene expression related to that could have evolved later, right? Similarly, if you look at the morphological diversity along the body axis, we have what's called morphological diversity within our own body, from head to toe, right? That morphological diversity is much later evolved compared to early animals. For example, earthworm, roundworm, if you see, there's hardly any morphological diversity along the body axis. You can make out which is head and tail only when it's moving, because it's direction in which it's moving, you call it this is head and that's other tail. But if it's a still worm, you cannot make out which is head, which is tail, right? So the first feature to evolve morphologically is the head versus the rest of the body, right? And then what has happened is, any new gene which has evolved by duplication and divergence, it is adding new features to the posterior part of the body, not to the anterior one, because if it suppresses the anterior features and trying to evolve something new, invariably it will have much more deleterious impact on the survival of the organism, because the head is the most important aspect. But what you can add new features is along the posterior axis. So the way Hox genes have evolved, you keep on adding more and more features along the posterior part of the body axis, from one end to the other end in that direction. And other ones, there must have been all kinds of evolution. What is selected, what is retained now is in this order in which they are functioning, anterior to the posterior. So if the position on the chromosome is, that's how you can correlate. So if this is the beginning one side, right, this is the, if the centromere is here, and Hox protein 1, 2, 3, 4, you can count how many Hoxes are there. And the one which is closest to the centromere would be the one which would be functional more anterior of the body, and one which is go away from the centromere towards the distal end of the chromosome are the ones which will be more and more posterior part of the body.
But then say, you mentioned that these duplication events happen much later. These duplication events, so say they happen much later during the diversification of animals, but then they could have happened, so say a gene is getting duplicated, it could have duplicated at anywhere in the gene.
Duplication, divergence, not all of them would get selected in the population, right? Some will get completely eliminated if they have a negative impact on the genes. What survives is what we see today.
Okay, thank you.
Can we relate this thing to loss of tail in our case?
Loss of tail?
Yeah.
That people have studied, very different ways. I'm not an expert on that one, but there are two kinds of evolutionary pathways related to morphological one, right? Development of new features, and then loss of existing features, right? Loss can be multiple reasons, right? Simply a gene is, corresponding gene is not properly expressed, they become rudimentary or they may not be required, then there is accumulation of mutations, and because there is no negative impact of loss of that particular one. And once the bivitality on one particular body, features and our behavior, walking behavior, is everything changed, it may not be, the tail may not be a vital organ. Once it's not a vital organ, mutations in it becomes either vestigial or can completely get eliminated, right? So there can be different reasons why it could have happened. I don't know very specific related to the absence of tail in you. But we do have tail bone still, suggesting that it must have been lost sometime during evolution. Okay, so I'll just leave with in another 20 minutes or so. So I started with some, because it's one cell to multiple cells, one same genome, giving rise to all kinds of morphological diversity within one organism's body. So we started saying that, is a spatiotemporal gene regulation and some aspect of cytoskeletal dynamics are the ones which together will form different types of cells and different types of tissues and organs and so forth, right? But is that really all about? Because, one is of course gene expression, right? And gene expression, let's say generates certain kinds of cellular features in terms of their shape, size, some internal dynamics, right? But how in a group of cells this sort of is, manifested? Is that always dependent on gene expression or some other, forces operating, is simply because these cells are in a group, right? So what kind of mechanical properties of cells are also important in understanding the development, right? You cannot reduce everything into only gene expression. Now, but interestingly, let's say, a subtle cellular identity like epithelial cells. Now, the two epithelial cells versus thousands of epithelial cells together will have a slightly different, not slightly different, developmental outcome, is simply because the interaction is much more complex, more forces are operating, from a variety of different directions, right? So those are the kind of questions one can ask. This is where quantitative and computational biology has to excel, because it's very difficult to do every one of these things using experimental method, right? And also there are certain kinds of our passive role versus active role, right? Let's say, gene expression is an active role because it's clearly programmed. Morphogen gradient, how it is initiated in Drosophila, I explained in great detail, and it's a reproducible and mechanism. It happens every time a new fly is born. Billions of flies are being born every week, and you see the same pattern getting reproduced. Same thing human too. Whatever the early events, they are reproduced with high degree of fidelity every time a new individual is born with hardly any defects. And so that's basically because is an actively gene expression patterns are regulated, what is expressed, what is not expressed, what should be repressed, what should be activated, and so forth. Then the question comes, what are the passive role of this? Let's say 10 cells versus 500 cells versus 5,000 cells is simply interacting with each other, and although there are a lot of dynamics in that part, the reason I use it passive is it may not be programmed, but it is an emergent property of large number of cells coming together. So what is actively regulated is how many cell division, let's say a group of cells have to go through to make this limb, let's say, right? And then the number of cell division is such that you will get, let's say, 1 billion cells. Once 1 billion cells are present in a field, how they sort of interact with each other? What is the emergent property of that? Will it be always like this kind of a hand? Can be a passive, or it can also have multiple active, other very active mechanisms may be present, right? Even if we talk about mechanical forces, are they also genetically programmed? Are they programmed, mechanical forces generating, right? If let's say I have an epithelial cell and it has certain dynamics, it has certain forces operating both internally and internally with help of actin, myosin complexes, externally because of interaction with other cells or extracellular matrix, whatever, right? And is this programmed, right? 1 cell, 2 cells, 10 cells is exactly the same way mechanical forces operate, then it should be programmed. Or is the mechanical forces an emergent property of a group of cells, depending on how many cells are interacting with each other, then you say it may not be always genetically programmed, right? So but this is something which one can experimentally test, which also can be, of course, modeled in any of their computational methods. I'll take one example of some of the recent work we have done in this area, which is very preliminary yet, but I'll sort of try to explain this. So again, the difference is wing and halter, right? Wing is a flat epithelial structure, halter is a bulbous structure, right? It's like this, and the flatness, you can see that these are called trichomes, the wing hairs, which are required for for proper control of the aerodynamics during flight, right? So when in the way it happens is when the insect is flying and these hairs are sort of in the direction of the flight, so that the air current that is coming will not be inhibitory to the flight, right? And in case of halter, it is a bulbous structure and much more densely packed hairs, and these are more sparsely arranged, right? But they are not directly involved in the flight; they are more balancing function during flight and also steering, how quickly you can change the direction of the flight and so forth. But the two the progenitors of both wing and halter is more or less identical epithelial cells, because if UBX is removed during larval development, what you get is a wing. UBX can be removed, there's something where Achishman asked the question, if you remove UBX early during halter specification, later during larval development, you can still get halter during development. But if UBX is removed much later, afterwards pupal development, you may not get the same phenotype. So there is also temporal requirement of UBX. Most importantly, in this context what we are discussing now, UBX, although it is expressed in the larvae, and the two have common progenitors, right? Because if you remove UBX, what you get is a wing, right? So that means starting with similar kind of progenitor cells, you can get two very different size and shape organs, and that size and shape determines function, so we will not worry about the function. Simply look at it from the structural point of view. The size and shape of these organs are so different, although starting from the same kind of epithelial cells, right? And I'll not get into the details of morphogenesis. That itself is a fantastic topic for anybody to study. There so much is done. It's also worth using this as a model system for you to learn computational method of looking at morphogenesis, right? So you start with some positional information. As you said, this is called secondary field. Primary field is the embryonic development. Secondary field is during adult development these structures are formed, right? And that's why it's called secondary field. Now the anterior and the posterior information and dorsal and ventral information, this is there from the early embryonic development, and the same thing is continued later during larval development for these adult tissues. It's a group of cells. This group of cells also more or less equivalent cells. They do not have any further morphological features or any specific the blueprint of what is going to be formed, but they are still divided into 4 groups, right? based on the axis information that is set up early during development, anterior, posterior and dorsoventral. So you can divide them into 4 quadrants, and each quadrant has certain identity. Although between the quadrants also there is no other morphological or biochemical identities clearly mentioned, right? But this is further elaborated over time using, because of cell-cell interaction and because of which new gene expression happens. And over the time they get, there is some kind of a blueprint is made in this green part of it. Don't worry about what is this, it contributes to some other parts of the body. It is basically the back of the insect. And this, the green part is the one which is going to form the actual wing, the flight appendage. And from this small group of cells, which will basically a single layer of epithelial cells, and during development they sort of start expanding and elongating, what you call an eversion. And this eversion is basically from group of these green cells start moving forward more distally away from the body, whereas this will remain with the main body of the insect, right? And this starts getting elongated from once a layer of cells, you get now 2 layers of cells, right? And that this kind of a structure is formed. And we know a lot about development of this. A lot of cell stretching happens, and it's like this. It's a single layer, but there is a middle layer of cells which have slightly different properties, and this properties is an emergent property of interaction between dorsal and ventral cells. This is, let's say, dorsal and this is ventral cell, and there's an imaginary boundary, and these boundary cells acquire new properties because of interaction between dorsal and ventral cells. And these cells with the ones which basically the end of the elongation. When this happens, when they are getting elongated, the dorsal and ventral cells get snapped together like this. So now you get a 2-layer structure from a single layer. Obviously, it's not simply which gene is expressed, how many times cell division happens. A lot of mechanical properties have to be changed during development to generate this kind of a structure, right? So it's well studied system. So what we try to do is to see what happens during halter development, given all of these things which is known during wing development, right? So these images are not so good. This is the wing, this is the halter. Early up to 3rd instar stages, you hardly see any major differences in the gene expression pattern between halter. There are some minor gene expression pattern. I could show you in the beginning. I mentioned that there's a gene called vestigial which is suppressed in the halter because halter will not make wings, and wing imaginary discs do express that gene expression. They both start as columnar cells. So these epithelial cells are column shaped cells, and at some point, the wing ones, they become the columnar to cuboidal. From cuboidal, then they become very flat squamous cells, and that's how you get this kind of a structure, right? Whereas in halter, they, at the stage when the wing cells are cuboidal, they still remain as columnar, and later they become cuboidal and then remain cuboidal even at the adult stage. These are all observations under microscope, which is sort of well known, right? So the kind of work we undertook, Dilsha is the student who did all this work. What are the initial constraints in terms of cell shape, height, size and tension, extracellular matrix practice, octomycin levels and one, and then some junctional properties which we have not studied. See, the idea is to get this kind of a structure versus this kind of a structure. Size, there are 2 reasons why size difference. One is the cells, if they are flattened, in the surface area increases, then they have a slightly different size, although shape is different, size also, whereas this is more bulbous. The second is the cell number. So we are not discussing cell number here, and we are only looking at the size and shape based on the individual cell properties, not the number of cells. Now, what could be the reasons why we get two different tissues starting from similar kind of group of cells? Could be any one of these things or a combination of this is the beginning of the study. So, first observation was made is you look at, these are all columnar cells. You can, with the help of good microscopy, you can see from the top, you can see from the side, you can see from the bottom, you can look at from all angles, right? And you can see what is the difference between the two groups of cells, wing versus halter, in terms of the cell shape, right? One is, she has shown that the length wise, the halter cells are shorter compared to the wing. Second is the surface area from the top when you look at it, halter cells are much broader compared to the wing, right? And this is the reason for that is typically the wing columnar cells at the one end, what called apical end, is they are more constricted like this, right? It's not like a proper cylinder. At one end it is constricted. That constriction is because of actomycin complex proteins which pushes the membrane, and that sort of makes this kind of a narrow constriction. Now, in case of halter, it is more flat like this, right? Though the increased length of wing cells, it could be simply because the membrane is protruding and constriction is formed because of the actomycin complex, and in the halter it may not be happening, right? This is the first observation. Obviously, you have to show whether actomycin complex is involved in this, and indeed, and without getting into the details, the wing cells have higher levels of actomycin complexes compared to the halter cells, suggesting that indeed the constriction is the reason. The one side it is, in wing cells it is constricted, in halter it is not constricted. Now, if this constriction is basically, it's like a stretching the membrane. If the membrane is stretched because actomycin complex at the cortical level, at the cortical end, you expect that the tension on the membrane is higher in case of where membrane is stretched versus where membrane is not stretched, right? So, you can do this kind of experiment these days by simply ablating membrane from one end and see how much is the recoil. Like, it's like you stretch a rubber string and cut it in the middle, and then you see that it gets snapped at the two ends on the opposite side, and this is called recoil. You can actually measure this. How much is the displacement, what is the recoil velocity, which will basically give you an indication of the cortical tension. That cortical tension is higher in wing compared to halter, right? So, obviously, for our eyes, we cannot perceive this. It's all very fast event, and you have to sort of do some measurements, and those measurements will clearly suggest that wing cells have higher cortical, recoil velocity and larger displacement compared to the halter one, right? So, I'll skip this in the interest of the time. So, at the same time, some earlier in collaboration with another group, my lab had shown that extracellular matrix plays an important role in wing and halter development, right? So, just to so what happens here is, I think yeah, I have this, right? So, I told you, right, there is in all epithelial cells, just outside there is a so-called extracellular matrix. This is a protein which is secreted by these epithelial cells just to keep some kind of a buffer zone around the cells. This is to protect the cells from external forces, right? During cell division or for any internal dynamics, every cell has to remain independent from other cells. Although it may be connected to other cells for as a part of a multicellular system with the help of cell adhesion. There are some as part of the ECM dynamics. Their individual identity as epithelial cells remain because of this extracellular matrix, right? This extracellular matrix cannot be static. If it is static, there can't be any morphogenesis, right? As if an epithelial cell remain only one particular size and shape if extracellular matrix is not remodeled. And extracellular matrix remodeling, Ramray Bhatt must have spoken about in the context of cancer, how extracellular matrix remodeling is important for metastasis and so forth. So in case of wing, what happens is early during development, when the eversion is happening, the two layers are coming closer to each other. This extracellular matrix, particularly the basal membrane, ECM, is degraded and the two come very close to each other. Otherwise, the cells will not be very close to each other. They are so close to each other such that the two membrane proteins, right, integrins on both sides, they come and sort of make, interlocking kind of system, like a Velcro, right? And in case of halter, there's no such ECM degradation. Why it doesn't have ECM degradation? ECM degradation requires certain kinds of, proteases. These proteases are the one which degrade the proteins in the ECM, and that helps in the ECM remodeling. And UBX, which is a master regulatory gene, but we had shown earlier that UBX inhibits these kind of proteases, which is required for degradation of A1. There are two, MMP1 and MMP2. MMP1 is the one which is required for first stage of ECM degradation. MMP2 is for the second time of ECM degradation. And UBX degrades MMP1, so there is no degradation of ECM. If ECM is not degraded, the dorsal and ventral layer also they are getting formed, they cannot come very close. If they cannot come close, they sort of remain apart like this. If it remain apart, it's still not very bulbous. You can still do parallel kind of thing, right? So now the question is, how do you get this bulbous shape if given all these observations? So we made these observations, right? One observation is the cortical tension. Second observation is the, ECM degradation, right? Just looking at these parameters, which we have observed early during embryonic development, early during larval development and later in the early pupal development. It is also, what you can do is, you can actually observe at a certain stage during development what happens in the pupal development and when the eversion is started. You can see here, in case of wing, the ECM is getting degraded as much as possible, and you can see that the two layers have come very close. You can see only one kind of a red line suggesting that the two layers have come closer. Whereas in the halter, the two red lines in between a lot of these, in ECM protein which is present, making this more bulbous structure. But it will not explain everything that's happening, right? What other factors are involved. So what Mandar Inamdar is a professor at IIT Bombay, is a civil engineer, but he's more interested in biological systems, and he modeled this. And he modeled only taking 2 or 3 parameters, which all have observations, right? Observational evidence. One is the cortical tension. Second is the forces operating from the ECM, and the timing of ECM degradation. One is early in case of wing and much later in case of heart. And then he simply modeled this, and you can see here in case of wing, because the two layers are zipped together and they is sort of coupled now. The dorsal ventral layer development is coupled, and then they are basically stretching laterally. That basically helps more and more flattening of the cells, leading to more flattened structure. In case of halter, so this is the observation, and this is the end result, and you'll see how with time it still increases in the lumen size, the volume of the between the two layers, but the layers, the dorsal and ventral layer cells are basically getting more and more separated in space, but their cell shape is not changed so much. Cell shape changes is because of the ECM degradation. In the absence of ECM degradation, the cell shape is not changed so much, because ECM is a very important factor. I told you, right? ECM is the one which protects this epithelial cells for any changes, because it sort of isolates the epithelial cells from any other forces to operate on those cells. And I'll not get into the details of all the mutant versions that he looked at it. So what this model suggests is, if you, whatever the observational parameters that we have seen, there also could be many other factors may be operating, which you may not have studied, which you may not have been able to observe, because there are certain constraints in observation itself, because they are so opaque after later during development, you not be able to even observe under microscope a living pupa, right? So, but then how do you even study what happens during development? So the modeling, what it helped is just take 3 or 4 parameters for which we have observational evidence and see whether those parameters are good enough to see the end result. Because we know what is in the disc level, we have some information at the early pupal stage, and what we know is the adult structure, because that is can be observed. So from in between what happens, it's a black box, and that's where the mathematical modeling has helped, because he could show that these 3 features, right, observational height, size, actinomycin, cell tension, whatever is, and ECM, just looking at these parameters, if you model, you can actually see that whatever, where is my, yeah, right. So what we have observed in the disc level, we can observe because it's amenable to all observation, experimental intervention. The pupal stage, partially you can observe early during development, only about 6 to 8 hours you can observe well, you can make some measurements too. But later we don't know what happens in the next close to about 72 hours. And what happens in that 72 hours to get these kind of structure is how he has modeled, suggesting that these 4 features are the most important features for attaining, for those tissues to attain specific size, shape during development. Yeah. By the way, this is normal wing, this is normal halter, this is mutant ones, that's why we have talked about it. So basically it suggests that these parameters are good enough to determine size and shape. That means some genetic programming of development generates certain kinds of forces, and those forces operate in a particular way and a very reproducible way to generate these kind of features in the developing wing and halter. I'll stop here. We can take some questions.
Thank you so much. So any questions?
So the primary function of UBX seems to be for wing development.
Seems to be for
Wing development. So
suppressing the wing development and specifying the halter. UBX function, yeah.
oh,
Hind wing, not hind wing, hind limb you can say. In case of Drosophila, hind limb is a pair of halter, not wing.
okay. Sorry, I misunderstood. Okay, so I have another question, maybe not very pertinent to what you've discussed, but I just want to have your opinion. So the case where you expressed eye in a Drosophila leg, so you mentioned that the eye is responsive to light, but I suppose it's not
There's
functional.
perception through that eye. That's what
Right, it's not functional. So for that you need to connect to the brain through
Right.
optic nerve, I suppose. Now I read some papers where people induced eye in the zebrafish stomach, and they were sort of claiming that the eye is functional. So apparently there is sort of an optic nerve connected to the brain. So I was just wondering, is it easier for the eye to, find a connection with the brain when it's induced in the stomach?
I don't know which is the latest one, I may not have read. But earlier, when the Drosophila work was done in late 90s and early 2000, many people tried in in zebrafish, in zebrafish as well as in frog xenopus, similar kind of experiments. And there also there was no physiological connection between the new eyes to the brain. It was only a local transformation of sulfate from one type of epithelial to eye cells. Eye cells have two types of cells, ectodermal epithelial cells and the neuronal cells. Neuronal cells are the photoreceptor cells. The photoreceptor cells function like photoreceptor cells, and epithelial cells or pigment cells function like pigment cells. But they are not connected to the brain, so there is no perception. But I don't know if something latest I have not read that. Anybody knows? Any others?
Okay, so
Thanks.
any more
Because
questions?
if you look at early during development, it's a coordinated, right? It's let's say we are made up of 50 trillion cells, right? It's not that end of 50 trillion cells, very different identities are specified. When 2 cells, already there are 2 identities. 4 cells, there are 4 identities, right? Like this, it keeps growing, and then at some point you will have, let's say, 100 cells with eye identity. Later it may become 100,000 cells with still eye identity, right? So the identities are specified early. And now if physiological connections had to be made between an eye in the abdomen to the brain, right, the all other intermediate cells should also have certain kinds of changes, saying that these neurons have to be connected, right? I doubt if perception is possible from such ectopic eyes. Sorry, one second, use the microphone. Mic, please.
So, can distance from brain is like also responsible for say non-functional?
It's not the question of physical distance, it's the whole the developmental mechanism, right? As I just mentioned, that every cell has certain identity as it's sort of in the position along the body axis. It may start with 1 cell at a time, then 2 cell, 4 cell, 8 cells, and so forth. But and each time there's some identities are taken up. That group of cells, one identity and other group of cells, even if they are close to each other, they do not interact. They are separated in using the help of, in the absence of compatible cell adhesion cell properties, they may not even interact with each other, right? They may not have they may interact in a very different way, like repelling interaction. In Drosophila, the wing disc, although it's one common single layer structure, the anterior cell and the posterior cells, they are separated, and there is a middle cell which called as border cells. They have certain specific properties. Only those cells can, one side it can have anterior, other side posterior. But if you take a posterior and put in anterior cell, it sort of gets repelled out from that place, right? It's simply because they cannot mix and remain there. So distance is really not so important. The same thing, neurons also, right? Neuron to neuron connection is a synaptic connection, or neuron to muscle connection. The compatibility is important. It's simply physical presence, distance is not always determines whether they make synaptic contacts or not.
Just one more question. So can I say that when say they are expressing some gene, they are expressing some genes somewhere around in, so it's like not functional. So it's just representing their like what, not representing, it's just their say eye. So we can just what do you say, like physiological change you see. So is there any experiment when they mutate with some functional gene, say somewhere else? So it's like you see that okay, it should be expressed somewhere else, but it is expressed here, and it is also showing the same functional what?
No. Okay. So if I understood correctly, you're saying, can we induce similar kind of physiological connection between 2 cells, but not necessarily in the normal conditions? There can be a completely 2 different positions, but you make them compatible in terms of physiological connection by gene expression pattern. Is that what you're asking?
Not exactly. So I'm like, so if you mutate, say for example leg or some for example, leg or some other part, some other limb, and if you express it somewhere else, it's like not physiologically. Are there functional properties like? Uh, so, I mean, like, can this show functional properties as in,
Yeah,
like,
yeah, I mean,
not just
I mean,
physiological?
that's what I'm telling you. During evolution, one can actually think of, morphological features. Sometimes it can be an existing morphological feature regained, and can have a new function also. But let's say in case of Drosophila UBX, right? You remove UBX, you get a wing. That wing is not flapping wing, right? It's not so functional. It has structurally it is like a perfect wing, like the front wing. There you'll have another pair of in the T3, the hind wing, identical, but there is no function to this simply because the corresponding muscles which required for generating the forces to flap the wing are not developed like in the second thoracic segment. Although UBX is removed. What UBX removal basically changes only certain epithelial structure but not the muscles. So in a way it's not functional wing. But if you look at this, I call it as a four-winged insect, but it is not really a functional wing, it's only a structural wing. I should have mentioned it. So that clearly suggests that you need more than one structure to get the function. Function is not simply structure is important of that particular tissue, but there is something else also has to come.
Can you say more about the connection thing as well?
Yeah, the multicellular organism thing is there are certain of autonomy to its cells, like neutrophil cells happily move around and keep eating bacteria, right? They have functional autonomy, right? At the same time, there are certain other type of organs or tissues, they don't have functional autonomy, although they have function is dependent on the structure, but their function is whether they get a neuronal input or whether they have a right muscle connection.
Thanks.
Okay, so any more questions? Okay, if not, let's thank the speaker once again.
Thank you.
So we'll break for tea now. Tea will be just outside the auditorium, and we will.
Just